Global Protein Production Harmonization, Alignment of the Protein Production from Discovery Research through Manufacturing by Brian Dwyer, April 2020. Executive Summary, Global Harmonization of a Pharmaceutical Company. Incremental steps towards global protein therapeutic production harmonization resulted in reduction in timeline by up to 7 years, which represented a 50% reduction in regards to the original state of the system. Reduce the number of full-time employees per department, and reduce the number of decision states by over 50%. In essence, we reduce the complexity of the system as well as shortening the time it takes to complete it. These refinements are primarily represented by the removal of waste, which is really about the MUDA in regards to the lean system. We removed built-in redundancies. This harmonization effort improves through the continuous focus on optimization, Kaizen. As this process is continuing, it's an active state versus a static state. Introduction to global protein production, original state of the system. So this is in regards to harmonization within a pharmaceutical company, and we're focusing on one aspect of that pharmaceutical company, which is the production of protein therapeutics. There are various stages. Early on it's called discovery, and later on, right before going to market, it goes out through a phase 3 clinical trial. There are various tools needed and formats needed at each stage. As you can imagine, producing a protein therapeutic and having it get out as a marketed drug is no easy task and takes various expertise and various ways of thinking to get it to the finish line. Even with good communication systems, separate divisions become focused on success within their own division. This is true, and you'll see through the harmonization we try to limit this phenomenon. Overall success of the entire system is limited due to this fact. So short-sightedness and lack of harmony and flattening across all divisions. Producing a global mindset and process will ensure efficiency and actualization of a non-limited success for the entire system. In essence, we're moving it above and beyond nested groups into the whole group. So what we see here is the initial state of the protein production system. Basically, what we have is a list of items that are needed for protein production. You need a construct. This construct contains the information of what protein you're going to make. That construct goes into a cell line, and that cell line is actually what produces the protein. Once the protein is produced by the cell, it needs to be purified. And then after purification, the protein needs to be characterized so that you actually have what you're expecting to have. And then it needs to be in a formulation, meaning in the final fluid or buffer that it needs to be in to go out and be sold in the marketplace. On the top, with a bunch of different colored bars, you see the various stages you go along throughout the pharmaceutical company. You have early discovery, you have discovery research, discovery therapeutics, to development, to clinical phase 1, and then I've grouped clinical phase 2 and 3 together. In the green box, you see that there's the market. I won't be talking about the market, just want you to know that it's another area in which you go into when you leave phase 3 clinical trials. Below it is to show the complexity around the management. Each of the subgroups has a director, and usually these directors report to a fewer number of vice presidents. And what you see by the bars are basically which division makes what decisions. So from the construct, what you see is early discovery will make their own construct. And when it moves to discovery research, they'll have their own construct. And then when it goes into discovery therapy, where you're actually going to make the protein, they're probably going to make their own construct. And then when they go to development, which angles the whole process towards clinical trials, they're going to probably use their own construct, something that they're used to working with. And that may carry through phase 1, but then there are critical decisions moving into phase 2 and 3 because the amount of protein that's needed goes way up, and there will be new considerations around the construct. So that's just to show you one thing where you have potentially 1, 2, 3, 4, 5 different constructs designed and made. And someone from each of these groups is going to be doing this, all of them trying to do a good job, all of them trying to produce something that's going to be useful for the phase that it's in. This can be true for the cell line, and you'll see the same phenomenon is true for purification, characterization, and formulation. In this slide, I just want to put these red lines in where I call them differentiation or significant differentiation. So minor for the small one, thicker for the big ones. So here is where I'd like to focus on where decision states are made. So you can imagine early discovery, there's decisions made when you go into discovery research, decisions that are made when you go discovery therapeutic. When you go in development, there's a fundamental shift in focus, which is towards clinic. So there's more significant decisions state at that point on various things, the construct of cell line, the protein purification and characterization and formulation. And again, when you go from phase 1 to phase 2, 3, there's a significant decisions that are needed to make because the cost for goods goes way up. So, and also you can see among these lines, you have different directors that need to communicate with each other, which may be facilitated by their independent vice presidents, but in the beginning, big decisions are actually carried through by one vice president to the other, obviously in concert with the directors that are below them. So this adds a level of complexity outside of just the normal workflow, this sort of the structured, nested nature of decision making, and the promotion of walls and separation and focus on internal optimization. So the harmonization is focused in both vertically and horizontally. So protein production is punctuated horizontally, which I just described, and it's complicated vertically. So we can try and, when we're doing harmonization, we basically want to alleviate both of these directions. So potential for lean is to remove waste. So there's a lot of the muda, which is going to be the waiting, over processing, overproduction, and over motion, really. All of this can be removed by harmonizing horizontally and vertically. You can just imagine. We want to focus on constant improvement as well, which would be a continuous focus of this process. Once we finish the harmonization and in a global sense, we want to keep this initiative rolling and empower the folks that are on doing the work to describe how to do it better, more efficiently. This is a total production management issue as well, because we want to talk about it in a comprehensive sense. And there is some complexity here that you cannot remove, but at least if you can pull things earlier, have various representatives from each of the major decision bodies vertically and horizontally meeting earlier, we can take care of a lot of this, and you'll see this as we move forward. The pull system is Kanban, so obviously this is in a slightly different manner than we're used to in regards to a Toyota manufacturing process. You're not going to run out of protein from phase 2, 3, and that's going to pull phase 1, which is going to pull development. It's not really working that way. But at least we don't pull people from the various horizontally connected divisions into a meeting unless they're needed for the next step, right? Or we want to pull them in earlier, and that's what I mean by pull, and carry that thing forward along the way towards the final step, which is the clinical phase trial 3. And there's a value stream mapping because the value really is in the sold product, which is at the end. But we want to make sure that the decisions we make early on contain that value as early as possible, meaning empowering that final acquisition and attainment of that value at the end, which again, what you'll see is the time frame. We're talking out almost 15 years of the original format. So it's a long-term horizon which people are not used to thinking. But if we can pull the people who are involved in clinical trial decisions, development decisions, discovery therapeutic decisions, and so on, as early discovery, we should be helping enable a final construct right off the bat that can carry through all horizontal movements and be useful at the end. This will get rid of a lot of waste and allow for a lot of reduction of complexity and cost. Another aspect that needs to be pointed out is the vertical aspect. And if we focus on the column under discovery therapy, we see a bunch of blue boxes. And that the blue is really assigned to that discovery therapy. And discovery therapy is where a lot of the protein purification, protein characterization expertise is housed. The best protein purification, best protein biochemists and biophysicists are housed in this division. So what you'll see as we move along to help with the vertical complexity is, for example, with the protein purification, you'll see more of the blue box expanding out horizontally, meaning the expertise of the discovery therapy folks will be broadened out. And it makes sense that you use the experts around protein purification in your harmonization process to inform the earlier processes in research, discovery research or early discovery, and even into development. This is an important distinction in that part of the pull process I want to use for this, which is perhaps slightly modified from traditional lean pull processes, is that you're going to pull from the experts into the directions you need them, and their processes will be broadened out horizontally and also be implemented vertically. In that sense, you have the appropriate expertise driving the change or the harmonization in the appropriate places and for the appropriate processes. And eventually you'll have harmonization horizontally and vertically that is most informed by the most appropriate intellectual backgrounds and functional backgrounds.